Okay, welcome back to Biochem 2280, McLaughlin edition. We're going to continue on with topic 14 today and hopefully make some progress on that. You should have seen announcements about room assignments for the midterm and about Q&As we're going to be having next week. So, pay attention to that, mark those down so you can get help if you need it. But certainly you want to know where you go to write the test. Okay. So in this part, we are going to continue on with our case study of FOXP2 and how we're trying to clone the DNA for this. And this reminded me of this comic here, where Calvin decided it's a good idea for Hobbes to cut his hair. How this works with a stuffed animal, I don't know, but we just go with it in the imagination. So he's asking him how he'd like it cut, and he tells him, and then Hobbes keeps suggesting that maybe it should be real short, in this very particular spot, right? And this is bad news, right? When you go to get your hair cut and you realize that the barber, the person cutting your hair, has made a mistake. That's not a good thing. So when we're cutting hair, we want it to be done exactly the right way. Same thing with DNA. If we're cutting DNA, we want it to be cut in exactly the right spots, and we'll talk about that. We'll get to that in today's lecture. Okay? So last time we left off, I was supposed to show this, but I was distracted by the throng of students from the first section asking me questions. So I'm actually going to be at this on Tuesday night. So if you're interested in that, future black physicians, and then Southeast Asian history, that's always good. Monday. Okay, so I've done my duty there. So now we came back to what we were talking about. So last time, this is our roadmap. We made some progress. We defined where which tissues express the RNA. We isolated the message. We synthesized cDNA. We specifically amplified our FOXP2 cDNA out of that, and we purified it using electrophoresis. Okay, that's what we're doing here. Now we want to try to put that into this weird thing called the E. coli expression plasmid to make a recombinant plasmid. So today we're going to be talking about that process and probably the transformation. Okay? So what are plasmids? What is this thing called a plasmid? You know that bacteria have chromosomes just like any other organism. It's got a genome. That's shown on the right here. This is a very large piece of circular DNA, double-stranded DNA. It's over 4 million base pairs in length in E. coli, and there's one copy of it per cell in a bacterium. Plasmid DNA are extrachromosomal DNA elements. They're independent of the chromosome. They replicate independently of the chromosome, and they're very much smaller than the chromosome, typically on the order of a few thousand bases, say 3,000, 4,000, 5,000, something like that. They are circular, tend to be circular, and they can exist in more than one copy per cell, and this depends on the particular type of plasmid that you have. Don't worry about details about that, but some plasmids are present in only very few copies per cell. Some are very abundant in a cell, maybe 100 copies in the cell. But these are separate from the chromosome, they're independent, and they replicate on their own. And because of that, it's a very handy way to introduce DNA into the bacterium that we want the bacterium to express. So these then, in this sense, become a so-called vector. A vector is something that transmits or transfers something or carries it from one place to another. That's the one use of the word vector. And so the plasmid DNAs can be a vector to introduce a foreign gene into E. coli. So we're going to talk about that now. Okay, so what features do plasmids have? This is a diagram from your book. And the first thing I want to call attention to is this over here, the so-called selectable marker. This is a gene that will allow you to determine which E. coli cells have your plasmid. And the one they're showing here is called AMP-R. That's short for ampicillin resistance. Ampicillin is an antibiotic that will kill bacteria that don't have any defense mechanism against it, and so we use strains in the lab that are not resistant to this antibiotic. except when we add a plasmid to them. So if the plasmid is present, then this gene here gets expressed by the bacterium, and that confers resistance to the antibiotic to the bacterium. So then we'll talk about this again later. This is a way you can select for all the bacteria that have the plasmid in it, right? Because if they don't have the plasmid, they'll get killed by the antibiotic, but if they have the plasmid, we can select for them, and they'll grow in the presence of the antibiotic. One other very important feature of the plasmid that they left off this figure, which I'm now adding, is an origin of replication. The plasmid itself, you want it to be able to be copied by the bacterium, so you need to have an origin of replication on it, because otherwise, you know, when the bacterium divides, you won't be able to spread the plasmid to the daughter cells. And so you always want to have an origin of replication on your plasmid. I've given an example of one here called E1. You don't need to worry about specifics of different types of origin of replication. Just be aware that you need one. And this is the same type of thing that Dr. Heinemann talked about when she's talking about replication. It's a space where the strands separate and replication begins, and then you replicate the plasmid just like you would replicate the chromosome. Okay? So you need to have one of those. And then the last feature here is indicated by these funny series of letters. And this is what we're calling a multiple cloning region. And what all that means is you have a bunch of these specific sequences here. Each one of these series of letters represents a different specific sequence, and they're identified by the so-called restriction endonucleases that will cut them. Okay, so that's what we've got here. Let me amplify a bit about that. Restriction endonucleases, what are they? They are endonucleases first, and just again as a refresher of your memory, what's an endonuclease? An endonuclease is an enzyme that will digest nucleic acids, particularly DNA. An endonuclease will cut in the middle of a DNA strand. Right? Endo is a prefix meaning inside. And you contrast that with exonucleases, which cut from the ends of DNA, right? So restriction endonucleases will cut DNA inside at a central location. They don't need an end to cut. And what's interesting about these restriction endonucleases in particular is they have a very specific recognition sequence that they cut at, and they're usually quite specific, and they won't cut DNA except at the sequences that they're specified for. Usually these sequences are palindromic sequences of 4, 6, or 8 base pairs as indicated. What do we mean by that? What's a palindrome? Well, in language, you may be aware that a palindrome is a word that reads the same forwards and backwards. So like radar, for example, it's got the same sequence of letters depending either way if you go forwards or backwards. That's a palindrome in language. In DNA, it's a little different. We define a palindrome as a sequence that reads the same from 5' to 3' on both strands. Right? So whichever strand you look at, the sequence 5' to 3' is the same. And so I've given you two examples here. GAA TTC is not palindromic in a language sense, but it is palindromic in a DNA sense, because if you look at the complementary strand, you see it also reads GAA TTC, 5' to 3'. You get that? Okay? So these are sort of mirror images of each other on the two strands. And the same thing over here, GAT ATC, and the other strand it also reads GAT ATC. So that's what we mean by palindromic sequences. So when we look at the multiple cloning region, each one of these represents a particular enzyme, a restriction enzyme. That's often what we abbreviate it to, restriction endonuclease and restriction enzyme. This is the same thing. So these are coding, or these are sequences on the DNA that are put together right one after the other that are recognition sequences for these different restriction enzymes. And we know about 300 different restriction enzymes by this point. And so we have a lot of flexibility there. And these then allow us to cut the DNA at very specific sequences, right? And if say, let me say a bit about naming. EcoR1, these restriction enzymes are named by the bacterium they come from. All restriction enzymes have their origin in bacteria. This is a bacterial sort of defense mechanism against foreign invasion. So EcoR1 is isolated from E. coli, and R, I think, refers to the strain, and then 1 means it was the first one that was isolated from E. coli. Okay, and so it has a very specific recognition sequence, and if that's the only site that has that sequence appears in the whole plasmid, then if you add EcoR1 to this plasmid, it'll make a specific cut right at that site, and we have a defined site where we can snip the DNA and cut it apart. Okay, so notice it makes double strand breaks as well. It'll cut both strands to make basically a double strand break in the DNA. Now, I just want to take a minute and talk about the palindrome, see if you got my explanation, and I'll put this up as a top hat question. All right, so I put 4 different sequences here. So these are not intended to be complementary to each other, right? These are 4 individual sequences. I want you to click on any one or multiple of them that you believe are palindromic in a DNA sense. I've only given you the one strand, so you have to imagine that the other strand is there. Typically, this is how we present DNA sequences. We give you one strand, the other strand is implied because of the Watson-Crick base pairing rules. Okay, a few more seconds. All right. Let's see what we got. Okay, so the bottom 2 attracted the most attention. This one attracted some attention. That is understandable because this is palindromic in a language sense, right? It reads the same forward and backwards. But in a DNA sense, it is not palindromic. And you easily see this if you have the sequence TAAAT, that's on one strand. Of course, the other strand would be ATTTTA, as those read different. On the 5' and, from 5' to 3' in the 2 strands. So this is not palindromic in a DNA sense. And likewise, this one over here, GTT, that's not anywhere near palindromic actually. It's AAC in the other direction, and that doesn't work. But the other 2 are, and in case you still don't believe me about this, those of you who are doubters, if that's your 5' to 3' on the one strand, you use the complementary base pairing rules to get this sequence on the other strand. And you see AAT, ATT is the same on both strands, and likewise for the 4th one there, same idea, GAC, GTC, which we have, okay? So that's just a little thing to hopefully solidify the palindromic nature, okay? So it turns out that they not only cut specific sequences, but they cut at a very specific spot in those sequences. And so these are the actual restriction cut sites for these 2 enzymes. EcoR1 cuts this sequence here, and EcoR5 cuts the one over here. These are Roman numerals, right? And it cuts very specifically between here, this G and the A, and it cuts at the corresponding site on the other strand, right? These are the same site on each strand. Okay, and so it's actually the result then, and for these 2 enzymes, is going to be 2 different types of ends. This one here will leave a 5' overhang, meaning that when the strands separate, you have 4 bases here on the 5' end of this top strand that are not base paired to anything. right? And this is, of course, the whole thing starts out double-stranded, but here we've got a 5' overhang, and we've got an identical complementary 5' overhang over here, okay? And we call these then sticky ends, because if you have another piece of DNA with the same type of overhang that's complementary, it will actually come in and you can get them to anneal to each other and they'll stick together. So we call them sticky ends, or we call this a 5' overhang. Okay? And this can be very useful when you're trying to stick pieces of DNA together to have these overhangs. And of course they'll only anneal if they have the right sequence to make the Watson-Crick base pairing work. And you can contrast that with the EcoR5, which cuts right in the middle of the recognition sequence, and these are called blunt ends, right? There's no overhang, it's just they end at the same spot. Yes, sir?
What happens if you have a 3' overhang?
What happens if you have a 3' overhang? Then you have a 3' overhang. The 3' end of the DNA is longer, okay? Speaking of which, that's a good idea. Let's make a question about that. Okay, so I'm giving you, you can see that up there. That little thing's a little annoying. Okay, so these are 3 other restriction endonucleases, and they cut in between the bases that are underlined. And you note that some of them, this one here, HINC2, as long as you have a pyrimidine and a purine, it doesn't matter which ones they are. So that's not quite palindromic, if you think about it. So that's just a little twist on it. But apart from that, they will cut in between the pyrimidine and purine, and this one cuts over here. So which one of these will give you a 3' overhang? And then it's just A, B, and C in that order, so I'll just do that. It might help you to draw it out if you need to. Okay, get your answers in and we will see what people think. Okay, so most of you thought it was the first one, PST1, and a few people thought it was BAM-H1. So if we have a look at that, this was the first one, and I said it cuts in between the underlying bases, which are here and here. And of course, it's always shown 5' to 3', right? That's the convention. Anytime you see a sequence, it's unless it's otherwise specified, if I showed you that, you would know it was 5' to 3' even without the labels. So when you do that and you get it to cut, you would end up with this being here. That's where the cut is. And then we have a G over on this side. And then the G is here, but then that got cut over there. So we end up having this over here. So there's 5', 3'. All right, and this is the 5' end, that's the 3' end over there. So this is a 3' overhang, right? Where you have, as the gentleman suggested, an overhang at the 3' end. So restriction enzymes can cut any of these different ways, and there's actually funky other enzymes that will cut in outside the sequence, but we're not going to worry about those in this course anyway. So I shouldn't have even said it. Anyway, so the rest of them you can work out yourself, and I'm going to be posting, I will After I've given the questions to the other section, I'll be allowing you to see them in Top Hat, or I'll be assigning them for you so you can look at them later. And you can have a look at that, but certainly the PST1 here gives you the 3' overhangs. Okay? So, but you can imagine then, this is really handy. So no more going to the hairdresser and getting cuts you don't want. This is very useful for us to very specifically cut DNA and be able to cut it and move it and paste it together using ligase. And so that's extremely valuable because before that we had no way, before discovering these things, we had no way of specifically moving around sections of DNA. Oh yeah, and then there's another question. You might want to think about how often, how likely is it to get a specific recognition sequence? So I actually made this, you actually have to fill in the number. I'll make it a little easier. Well, no, I won't. If you caught it, I had some choices, but I guess I decided to make this just a random number. So, what is the probability, if you have a random 4 base pair sequence, that it's going to be a recognition site for this enzyme here, HEY3, and this is its recognition site? One in how many? Okay, make your guesses. Okay, let's see where we go. Look. Oh, there's one that people like, 256. This is an interesting suggestion. Be curious to know how that works mathematically. Right. All right, so how would you possibly want to approach this question? Well, we're assuming here then that the probability of any base being any one of the 4 bases is 1 in 4. That's sort of a baseline assumption that's necessary to answer this question, so that is a bit of an underlying assumption. But if we just had, let's just take one base. What if I said to you, what are the chances that any random base you look at in a sequence is G? You would say the chances is 1 in 4, she says, because you've got 4 different possibilities. So we've got 4 different sites in this recognition sequence. The odds of this one being a G is 1 in 4. And of course the odds of this one being a G is also 1 in 4, assuming an equal distribution. The odds of this one being a C is, guess what, 1 in 4 and 1 in 4. So then this takes you maybe back to mathematics. I did this in high school. I'm not sure where you were exposed to this. How would you know how many probabilities there are? Do you add them together? What do? You actually multiply them together, because the odds of this one being a G is 1 in 4, and this being a G is 1 in 4. That means the odds of them being G and G is 1 in 16. There's 16 possible combinations. Only one of them is a double G, right? And then you add this one, then it becomes 16 times 4, which is 64, and then that times 4 gives you 256. So that was the majority answer, so good on you, those of you who were able to figure that out, right? So that means that if you have a short sequence like this, the odds of getting that are it's going to be fairly common. If we think our plasmid is about 3,000 bases, or base pairs, then we'd expect to see this sequence occurring a number of times. But as you increase the length of the site, of course, it becomes more and more rare. And such as a 6-base cutter, if you could quickly do the math, will be about 1 in, I think it's 4,096 or something like that. Okay, so then that might occur only once in our plasmid. And so those are the ones that are actually going to be most valuable to us. And these things were so valuable that they gave a Nobel Prize out for the people who discovered them and figured out how to use them. And these three gentlemen shared this. This was, it's hard to stress how important this was in the early days of molecular biology, the ability to specifically cut DNA and mix it and match it together. This made it a whole lot easier to manipulate genetic material. Okay, so that was worth a Nobel Prize. Okay, so then we're going to use that in our cloning procedure. We're going to, and the way you do that is you pick a site that is common within your plasmid and is on the foreign DNA that you want to insert, and you cut with those to give you complementary ends, and then you anneal those together. As I said, the sticky ends can come together, and then you use DNA ligase to ligate that together. And a key thing to know then about DNA ligase is you need a phosphate on the 5' end for this to work. I don't know if Dr. Heinemann talked about this or not, but this is true. It's true for during replication and so on. It's also true here. The ligase needs the 5' end and the 5' phosphate. Okay, so this is our strategy then for getting our copy of the cDNA copy of the message for FOXP2 into our plasmid. And you might think, wow, what are the odds that you're going to have one of these sites here that's also on both ends of your message from the organism? Well, the odds will be very, very small that you're going to be lucky enough to just happen to have restriction enzymes that are restriction sites that are perfectly aligned to allow this to happen. So this is where some manipulation comes into play. So we can use PCR to introduce sites, restriction cut sites, on the ends of our cDNA. And if you remember at the very end of last class, I talked about introducing mutations in the DNA, and I said you can make a mismatch. And in a mismatch on your primer to the template, and as long as the 3' end anneals, it'll get synthesized, and then when you do the PCR and it gets synthesized again, it'll use that improperly matched base, and it'll make that the template for the new strand. And as PCR goes on, you amplify the mismatch. Well, you can put pretty much anything you want at the 5' end within reason. You can't be too long, but you can make a large section, relatively large, like 10 bases on the end of the 5' end of your primer that don't match the template, and that's okay. As long as the 3' end anneals, it can be extended, and then when you synthesize in the other direction, it's going to copy whatever that stuff is that you added to the 5' end. So that could be how you could introduce, in this case, a BAMH1 cut site, which is GGA-TCC. And that wouldn't exist in your FOXP2 cDNA, but now you've added it in. Okay, that's a clever way to use PCR. Another thing you can do, you can just make little tiny bits. You can synthesize double-stranded DNA that has that restriction site in it, and then you can use ligase just to join those ends together. So it's that you could just stick those onto the end using ligase. And these pointers are driving me a little nuts, I have to say. You can take a little linker, it's called linker DNA, you just stick it on the end. So those are two ways you can introduce restriction enzyme cut sites of interest onto your insert, okay? And the plasmid is good because those have been engineered in there already. You get the plasmid and it's got a bunch of those sites all in a row. You pick your favorite one and you put them in. Okay, so the idea would be you cut both of these with the same enzyme, and it has complementary sticky ends, and under the right conditions, you can get them to anneal to each other. I sort of drew this as an oval just so I could make the figure look okay. Get the sequence in like this way. This could be round, so I hope that doesn't throw anybody off. And then you add DNA ligase, and it seals the nicks in the backbone, and you get your recombinant plasmid. Okay? It's pretty cool. Cut DNA from one spot, stick it in another. Yeah?
So plasmids are about 3,000 base pairs long, right?
Right.
So does that sort of restrict what kind of DNA you can put into them, like a long
Okay, so
protein you couldn't do?
he's basically like, how big can the insert be? Because plasmids tend to be fairly small. And if you have a really huge piece of DNA, it becomes harder and harder to ligate that into a plasmid. But many plasmids can accept fragments of considerable size. So you could even double the size of the plasmid this way. Or even basically, typically, I mean, the bigger they are, the harder they are to transform and later. But you can have plasmids of 8 or 10,000 base pairs, and then you have artificial chromosomes, which are even bigger. But as far as plasmids go, they tend to top out around 10,000 base pairs. But you could actually put in a fairly decent sized chunk of DNA in here. But yes, he's absolutely right. If you get too big, then it becomes harder and harder to make it work. Okay. The other thing you'll notice is this way, if we do this, both ends of this are identical, and so you can't really control whether the plasmid is going to go in that way or the other way, right? Because the ends are the same. So one way you could control that is to use two different enzymes on the two different ends that have different overhangs, and therefore you could introduce it specifically in a specific orientation into your plasmid. So I hope that makes sense. Basically, you don't have to use the same site. There's a bunch of those restriction enzyme sites all in a row, so you can pick two of them that you like and use those to have unique ends. So instead of having the same end here, you could use this one here and then have another one cut here with a different overhang, and you could do the same thing on your cDNA, and you could put it in with a specific orientation. So, but for simplicity, I'm just showing you the same enzyme on this slide. Okay, so that's how we make recombinant. So that's the word recombinant just means we've combined DNA from two different sources into the same double helix. Okay, that's just what recombinant means. Okay, so now we've made our recombinant plasmid, we've got our gene of interest, now we want to introduce it into E. coli. That's the next step, and we call this transformation. And transformation is just that. It's the process by which we induce the bacterial cells to take up the foreign DNA from the plasmid. And bacteria will do this sort of on their own. This is how antibiotic resistant genes get transferred from bacteria one to another. But they do it under certain circumstances sort of on their own time. But we want to make them do it. And so we have worked out just by empirical experimentation conditions that we can treat the cells under to make them competent. And when we say competent, all we mean is a competent cell is a cell that's able to take up external DNA. So that's just, when you hear that word, a competent E. coli cells, it just means they're primed and able to take up foreign DNA. And chemical treatment, there's different protocols that are out there. A lot of them involve using divalent cations like calcium. That's pretty common to treat them with calcium and spin them down, and you do some different treatments. Don't worry about the details. If you take my third-year lab course next year, we'll do that. Okay? But the point is, we can treat the cells in certain ways with chemicals to make them competent. Then they're able to take up the exogenous DNA. So then you incubate the competent cells in the presence of the plasmid, and you heat them, and you do some special treatments. And if you're lucky, some of the cells will take up your plasmid. Not all of them, okay, because they're doing their own thing, but we can induce them and trick them to try to get them to take up the DNA. The process by which this happens is a little undefined at present. There must be some way in which the bacteria create pores so the DNA can get across the membranes, but at some rate they will get in. So now I'm showing you the cells, and some of them have taken up the plasmid, and they're said to be transformed. That's great. But the problem is now we have a collection of cells, some of which have our plasmid, some of which don't. So we need a way to select for the cells that have the plasmid, so that's where that selectable marker comes in. We can plate them on agar as just a solid support and with some nutrients in there, anything that a bacterium likes to be able to grow. And you plate them after you transform them, and you let them grow overnight. And in the morning you come in and you hope to see this. And each one of these little dots represents a colony of bacteria. And if you've taken microbiology, this should be very familiar to you. Each one of these little colonies represents a single cell that was landed on the agar, and then as we incubated at 37 overnight, it reproduced and grew, and out of that single cell becomes a whole colony of bacteria. But these are all identical. They should be pretty much identical to each other. And the cool thing about this is you can pour the plates and put the antibiotic in there such that any cell that does not have your plasmid should die because it's not resistant to the antibiotic. And these cells that have grown are the ones that have taken up the plasmid because now they have that gene that allows them to be resistant to the antibiotic. And then this way we can select for the cells that have taken up the plasmid. At least we know that these cells have antibiotic resistance in them. And this actually is a plate that I took early in, I think, March of this year from my third year students. Somebody made this plate. Okay? So, here's a question for you. Do all bacteria on that plate I just showed you contain a plasmid with the intended insert? Remember, we cut the plasmid, we cut the cDNA, and we try to ligate them together. And then we put those into the bacterium. What do you think? Does every bacterium on that plate contain the insert as we intended? I'll give you a little bit more time. This is just, pick one. Only got two choices. Okay, so most people think not. That's good because nothing in life is ever perfect, right? And you know this from experience. So even though we do our best to make sure this works, there are different things that can go wrong. For example, the plasmid, once you've cut it, could just re-anneal and be ligated together without the insert. That could happen at some frequency, in which case the insert, you'd have a complete plasmid, but you wouldn't have the insert. Or if you've been sloppy, there might be some contaminating DNA around that somehow got in there and caused the plasmid to recircularize, and therefore it wouldn't contain the insert. But one important thing that I actually forgot to mention is at this stage, the bacteria will tend only to take up circular DNA. Okay, so DNA that has not been annealed and remade into a circular piece of DNA, it's much, much harder to transform that into bacteria. So for the most part, the bacteria is only going to take up circular DNA. Well, in fact, the DNA might get in, but if it's not circular, the bacterium will recognize it as foreign and it'll chew it up. Okay? But circular DNA it recognizes as sort of normal, so it'll like that. Okay? So basically, you don't tend to get linear fragments in. So you must have had some sort of ligation to get the thing in there, but it doesn't necessarily mean that the insert is present. So we need ways then to verify that the clones that we've isolated on our plate actually have the insert that we want. Okay? At each step, we got to make sure we have what we're looking for. There's different ways to do this. Okay, and I've sort of shown in a picture form 4 different ways you could go about this. In each case, you pick off a colony from that plate, the transformation plate, and you make up a liquid culture, and then you isolate the plasmid DNA out of those bacteria. And we have well-characterized techniques or methods to do this. This is pretty easy in the lab to do, and like I said, in 3rd year in my biochem lab, you will do this if you decide to take that course. You isolate the plasmid DNA, and then we can test it in different ways to see if our insert's there. One way would be hybridization. As I already mentioned, you could design a probe that's specific to your insert and see, does it anneal to my plasmid? And if it does, that's some evidence that your insert is actually there, right? Well, likewise, you could use PCR. You could design primers that are complementary to the plasmid itself and have them pointing in and try to amplify the region that should have your insert. And then you can see how big that fragment is. And if it's the right size, then that's some good evidence that your insert is in there. But in each of these cases, that's indirect evidence. It doesn't tell you, like the hybridization says, okay, that part of the insert's there, but maybe something weird happened and the rest of your insert got lost somehow. So hybridization, that's not foolproof. PCR, likewise, if you just amplify it and look for the size of the fragment, all you know is that there's a fragment of that size in there. You're not 100% sure that it's actually the insert you want. Okay? Another way to do this would be restriction mapping. That's where you use these restriction endonuclease cut sites and you look for a fragment pattern that matches the size you predict if your insert was present. And then sequencing is the last method. This is actually the most reliable method, but I do want to talk just for a minute about restriction mapping because it is a sort of an easy and quick way that you can check for your insert. And so how does this work? Well, we have a defined sequence of our plasmid. We've got a defined sequence that we wanted to put in, and you can look for sites on the plasmid and possibly in the insert that will be different. Give you different size fragments if you cut the plasmid with those restriction enzymes. And so, for example, let's suppose our original plasmid was 3,000 base pairs, and it has 2 EcoR1 sites separated by 500 bases here. And then we use another enzyme to put in our insert, which is blue here. And let's say the insert is 1,000 base pairs. And so we expect then the whole plasmid to be 4,000 with the blue insert. And let's, I'm imagining we've introduced a different cut site for this enzyme NCO1 here. And I've shown you then the expected sizes or the distances between these cut sites. Okay, do you see what's going on here? So what would happen if you digested your plasmid preparation with these 3 enzymes? So that is the next question. So you isolate DNA from 3 different colonies, A, B, and C, and you digest with EcoR1 and NCO1 and run in a gel. Which colony, and this is the results of the gel here. This is your electrophoresis, and I've shown you the size of the fragments on here, or at least these are the standards that you run. This is our expected recombinant plasmid. So if you were to digest with these 2 enzymes and assume you get complete digestion, which one of these patterns corresponds to what you would hope to see if your insert was actually there? Okay, a few more seconds. close it down. Look at our responses. So most people went for C. And so if you just look at and predict what you should see. If everything cuts properly, you get a 500 base pair piece and a 1,000 base pair piece, and then the rest of here must be 2,500, and that's exactly what we see in lane C here, about 2,500 fragment, 1,000 to 500. This would be 4,000, that would be like the whole thing if it only got cut once, and this would be like 25 and 5, that would be some other weird thing. So basically, you can do a quick screen this way to see. You pick a bunch of colonies and you screen them this way and see which ones might even possibly have my insert, because some of them won't, and you don't want to waste time looking at those. You would want to spend time on the ones that are giving you the sizes that you want to see. And then just as an aside, I assume that there's some way you can see the bands on the gel. How do you do that? I just wanted to call attention to this. You can run the gel, and then you add this DNA binding dye. One that we use is ethidium bromide. There are other examples. These are just fluorescent molecules, and they tend to work by sliding in between the base pairs of the DNA. And they tend to be not fluorescent when they're just in solution, or not very fluorescent, but when they get into the DNA, then they become fluorescent. So then you can expose your gel to a UV light, which is what so sort of like a purple, like a black light box. And then wherever the ethidium bromide is bound to DNA, you see a pink signal there. These are some standards, presumably that were run. I got this from Wikipedia. But then you have different bands here representing DNA fragments of different sizes. That's a very common way to identify your samples on the gel. Alternatively, you could actually, if you wanted to, radio label your fragments with p32 and expose to X-ray film. This tends to work if your DNA is present in such small quantities that it's hard to see by a dye method. You could try to label it and see specifically where it is using radioactivity. That tends to be more sensitive. Okay? So that's restriction mapping, but these three on the left have the drawback that they're not really specific in that you have a piece of the right size, but you don't know exactly what it is. So if you really want to verify your clone has the insert you want, you need to do DNA sequencing and look exactly at the sequence of your plasmid to absolutely make sure that it is the sequence that you intended to construct. Okay, and so that's what we're going to get into next week, is how do we sequence the DNA. And so I'll describe a couple methods to do that. So have a good weekend, and we'll see you on Tuesday.